The purpose of the investigation of deuterium depleted water was to see if it can produce any radioprotective effects. There's a little story behind this. I was studying for the PhD the radioprotection. And the idea was that I had to find a substance which hasn't been tested yet, which might have a radioprotective effect. And at a certain point, I was able to read a paper concerning the so-called radiomimetic effects of heavy water. It means prolonged administration of heavy water would produce effects similar to those produced by ionizing radiation. So I said, if there is such a tool like the deuterium depleted water, why don't I try to use it to see if it might protect against radiation? For the very beginning, there's a few things that have to be remembered concerning the effects of ionizing radiation. How does radiation kills? It kills by producing what you call water radiolysis. So it breaks down water and produces oxidative products that further will act on the usually cellular membrane of cells and will produce various deleterious effects. Of course, molecular lesions on various enzymatic systems of the cell or of the tissue or of the organism. DNA lesions, strand lesions, which are responsive for the mutational effects, responsive for the neoplastic effects of ionizing radiation. Immune suppression, which everybody knows, it will destroy all fast proliferating tissues in the organism, thus producing immune suppression by suppressing the bone marrow, the hematogenic bone marrow, and the stem cells which out of which the immune cells arise. And hematogenic bone marrow suppression, which will produce subsequent anemia, lymphopenia, thrombopenia, and whatever other accidents might produce. And finally, the so-called radio-induced apoptosis. So, if we had this tool, we know that the normal ratio is 140-45 parts per million. The alteration can go towards the increase, producing the heavy waters, or towards the depletion, and this would produce some biological effects. So if there are any biological effects, we know that radiation induces death by the so-called hematological syndrome, by pancytopenia, so reduction of all cells in the blood, and by immune suppression. And it also produced by the so-called gastrointestinal syndrome. Gastrointestinal mucosa is the fastest developing, the fastest proliferating tissue in the organism. So because of the radiation, it's going to die, it's going to slow off, and it's going to produce mucus atrophy, ulceration, fistulae, infections, which further are going to kill the experienced animal or the person which gets irradiated. So let's see how any radioprotective effects can be seen. We are going to investigate if there is any antioxidant mechanism. So we have tried to test the antioxidant enzyme activity. We have tried to see if there is any lipid peroxidation or increased or decreased lipid peroxidation. And also we would see if there is any stimulation of cell proliferation which will help the organism to replenish its depleted cells after the radiation. And this would produce the immune stimulating actions, and we could test the immune stimulating actions by immunologic screening, by immune modulation evaluation, and by inducing experimental infections in animals, irradiated or not. And also, if we want to see if there is any stimulation of cell proliferation by the replacement of the normal culture media with culture media reconstituted with deuterium depleted water, we could see if there is any stimulation of cultured cells, and we use splenic immune cells in culture, and we also tried to test some membrane mechanisms which might have been involved in the proliferation. What have we done? So first, let's see if there is any variation, true variation, in organisms that have received deuterium depleted water. Now the literature says that after 2 weeks of continuous administration of water, the whole amount of water in the organism is going to be exchanged. So it means we have to give at least 2 weeks, 15 days, maybe 20 days of water in order to produce the depletive effect. So what did we do? We gave to rats for a long time, and then we sacrificed those rats, and we took blood, we took samples from muscle, samples from liver, and so on. And then we distilled all the water from those tissues, and we tested by mass spectrometry the content in deuterium of the biological fluids. This was made in several opportunities in the university, in the physics departments of the university in Iași in Romania. And what have we observed? We have observed that no matter how long we gave the water, the deuterium depleted water, 30 ppm, no matter how long we gave it, we could not deplete the deuterium less than around 90 ppms. So this is the control, yeah, the normal concentration, around 144-45 parts per million. And after receiving 2-3 weeks of water exclusively, this water, we could reduce the concentration in the muscle and in the liver for about 90 parts per million, which is a good effect. But it also means that we cannot reduce very much the deuterium concentration in the organism. Now, the radiation survival test used 380 animals, Swiss male mice, during 2 years in several stages. So they usually received exclusively dry chow and then deuterium depleted water or distilled water ad libitum. So they drank as much as they wanted. The groups received 27 to 30 parts per million compared to the control groups with normal water. Nutrition and hydration for minimally 14 days before irradiations. We had 2 test groups. So the one group that received deuterium depleted water before and after irradiation, and receiving deuterium depleted water only before irradiation. And after, we did not give it anymore. And we also tried to do a third group, so receiving deuterium depleted only after irradiation, but this had no effects. Total body irradiation was made with gamma radiation from a cobalt device, and also we made a control group using a so-called chemical radiomimetic, so a chemical substance which acts as the radiation. It's called hydrochloric embihine, and it's a nitrogen mustard derivative. Everybody knows what is nitrogen mustard, a very highly poisonous substance. Radiation doses varied between 6.5 to 9.5 gray, and this is the LD50 for mice. Yeah? And also we used auxiliary control groups using the traditional radioprotector, which is called amifostine. It's a sulfhydric derivative which is used now. What have we observed? We have a true statistically significant survival for those groups. You can see here, yeah, and these are the probits for the survival. So we had a 55 average survival after the radiation in mice, and then we have an 80% and 78% in group 1 and group 2. So it means that giving deuterium depleted water at least 2 weeks before irradiation would increase this survival. And also we have used several groups combining this with amifostine. Yeah? So you can see here it has received deuterium depleted water, amifostine, deuterium depleted water, and you can see that the combination with the chemical radioprotector does not produce any significant effects. This means there is no additive or synergistic effects, not even